Hey guys. Hope those of you that celebrate Easter had a good one yesterday. I know on my end, the Easter Bunny did not come 100% through. He was having things shipped and apparently they weren't priority items. So a lot of little stories. All right. Well, as I said to the earlier group, hope you all had a nice Easter if you're celebrating and Passover. I think saw a lot of Facebook posts that everybody was hunkered down with family. So hopefully that was great and lots of candy chocolates to be had. So today I'm going to finish up our landscape lecture. There were only, a couple more slides. Do a little bit of review on some of those major concepts, and then I'm going to transition over to ecosystems. So we're moving up our ladder in ecology again, moving on to ecosystems, which brings in abiotic factors. So we've got our community, organisms, plant, animals in same space and time. And then we're seeing how they're using energy and how they survive, die, move across the landscape, die in other locations, and the energy in their bodies returns, into the soil and becomes fodder for new things to grow. So we're going to be thinking about how energy in the form of organisms, plants and animals, can be distributed across our patches, like we talked about in our landscape lecture, and how that energy can flow as these animals and plants, the plants may be dispersing, are moving across that landscape and shifting over time. So that's what we'll do. Just a few kind of maintenance things at the start. Lab this week is microplastics. So it will be a much briefer lab. we have worked together, Dr. Aldred, Kirsten, our TA, and I made a Spark timeline video type thing for you. Kirsten went out to City Beach and did the field survey, so she'll demonstrate you know, the collection process that you guys would have been doing this week. And then I went into the lab, donned my white coat, and showed you what we would be doing next week. So other than touching base, I don't think we're going to have much to say for next week's lab, so it should be even more brief than this week. Let me see what, we've got a chat question. Okay, thanks, Isabella. So any questions on the lab? I'm going to share my screen just so you guys can see what I'm talking about. Can everyone see the beach?
Yeah.
Okay. So kind of a crummy day for the Monday lab to be going out there anyway, so maybe we have a win here. But there's learning objectives. We'll talk a little bit about microplastics, how they're a concern. You know, you're probably aware of the macroplastics, the big gyres that are out in the marine oceans, but we've got, you know, plastics on the finer scale in our water bodies all over the place, in the rivers and streams, as well as in the lakes and ponds. So we'll talk about how they're kind of like sponges for any kind of harsh stuff that's in the landscape. Imagine little plastics that are floating near a dock, and then, you know, the gas from boats is there, and then they absorb it, and then it gets uptaken by fish and by animals. So we've talked about, I think, biomagnification and accumulation. We are inhaling this stuff as well. And so here's Kirsten. She's gonna give you a quick video on what it is that we're doing. She'll talk to you guys about our beaches that we would have investigated. We would have been breaking up and going to a couple different beaches, some northern ones, Point of Roche and City Beach, and then we would have been down at Port Kent and Ausable Point down by where I live. That's the rack line where you would be doing surveys, kind of the high tide line. And then the plastics. I went out to my beaches and got some pictures of it. So you'll get a chance to kind of look through these videos. These are macroplastics I collected, a shotgun shell. Sig butts, those are the big ones. And then here are the videos of me kind of doing the work in the lab that you guys would have been doing next week by just taking your sample and adding a lot of salt water to it. And that should density separate those lighter plastics. And so you'll get a chance to see the stuff under the scope, all the things that I found, and then learn about some of the research we've been doing in our lab. So this semester we've been testing out a new technique, Nile Red, so I included that down there. We've also been doing some greenhouse work, planting these plastics, planting corn plants in pots that have macro and microplastics, and also that have a fertilizer that comes from the bacterial mats from treated wastewater. So, you know, in an effort to be kind of sustainable and recycle some of that stuff, they've been, you know, treating it to remove heavy metals, making the harmful bacterial inert, grinding it up, and then, you know, pelletizing it and selling it over the counter. But it comes from wastewater treatment. And so you can imagine if particles are coming in, you know, through wastewater, that there's a possibility that it's going to be, you know, in those bags, and then you spread it on your lawn, and then it ultimately will run off or get into the groundwater and have implications. So anyways, you're going to get a chance to look through that and learn about the plastic crisis that we have ongoing. And that'll be this week and next week's lab. So again, it should be briefer, and we'll have to modify our assignment likely, but stay tuned on that. And I think you guys also have your deadline on your revision. So hopefully that's going okay, the optimal foraging lab report. We do have a test coming up on Friday. I've mentioned this before. It runs from the predation lecture on through the landscape ecology lecture. I think we're going to be good on time, so I'm going to probably devote a good portion of Wednesday's class to review for you guys. Okay, so we'll be able to do some review, and it'll be similar to the format that I had the last test. So I'll be putting it up on Moodle. You'll have the hour once you commit, but you have probably a little bit, you know, longer. I think I gave you like a day and into another day to complete that, okay? So just be ready, make sure you're preparing like you would a regular test, so you know, you're not flipping through trying to find the answers as you're taking it, you know, then you run short on time. So make sure you prepare as if, you know, you're taking it in class and do the best that you can, okay? I have gone through the up-to-date community homeworks, giving you guys some feedback on that for those that just submitted that. So for anyone who hasn't, I'll be sending out a reminder email. Really, really important for you guys to practice that community homework because you will definitely be seeing it on the test, okay? All right, so plan on some review on Wednesday and I'll get through our lectures today. All right, so I'm gonna switch back over to landscape ecology. And again, with landscape ecology, some of the key, let me know if you guys cannot see this PowerPoint, some of those key things we're thinking about addressing questions at different scales, fine scale, moderate to coarse scale. And you know, our experiments are done very fine scale. We have short lives, nothing is often long term. And we address those questions, you know, with a very pinpoint, you know, methodology, right? But ideally, we want to get that kind of level of precision, and we also want to be able to scale it up so that we can address things with realism. And you know, we've got all these technologies now. We've got the field that's kind of moving in terms of you know, changing paradigms and concepts that we can apply to seeing how things change over time in these patches. And there were some terms that I wanted you guys to be familiar with. You know, we had talked about scale and how, you know, certain questions, when we look finely, you know, competition might be important or predation might be important. Those, you know, species interactions could be important at that community level. But then when we scale it up to maybe the landscape level, it could be that there's some abiotic factor that becomes more important in terms of the dynamics that we see. And the terms, so very important for you to commit these to memory. Again, put a star next to that. This is, you know, good stuff for a multiple choice, I can imagine. But the term that probably would be trickiest for you guys to remember would be contagion. So just think of the arrangement of puzzle pieces. You know, imagine all these dark, you know, green ones that are forest all clustered together. And then thinking about the shape of your patch. The shape of your patch is really important because it'll dictate the amount of edge versus core, okay? And so if you have a more rectangular shaped patch, then you're gonna have less core, more edge, and there's all those abiotic factors that are higher risk. Is somebody just entering maybe that needs to mute? I'm just hearing a little bit of feedback. One good example is the fire that was very large scale at Yellowstone. And what it did is it burned differently, different extents, different intensities in certain areas, and different times too. And so you've got a patchwork mosaic of habitat that's regenerating. Some are earlier succession, some are later succession, some were spared and didn't get burned. And so you can see that that heterogeneity would be really great in, you know, helping for predator and prey to persist on the landscape. Remember, that was one of the big things we talked about. We mentioned not all organisms are going to respond to edge the same way. So if you reduce the area of your, you know, fragment or your patch, and it's smaller and smaller, so less core, more edge, that is great for things like our skunks and our, you know, mesopredators, the raccoons, right? But it's not good for those ground nesting birds. We mentioned size of islands become very important in terms of how many species you can accommodate. And so the larger your island, think of the niche idea, that you'd have more resources available, you'd have, you know, more dens, more nests and all that stuff. And we see that with lizards across these real islands, according to size. And when we cover, you know, that species specific response to habitat patch size, remember, not all of them behave the same way. So it was okay for us to have small patches and see a lot of robins and catbirds. They do okay with that. They're okay on the edge, with the edge. Maybe they've got preferred food that is, you know, something that does well when there's more edge versus core. We don't see that. It's kind of the opposite effect with warblers, and they're a little bit more sensitive. We find them in the conifer forests of New Hampshire and up where we are, and in New England. And then just some of them, they're fine wherever. It doesn't matter if it's a small patch or a medium patch or a large patch. So the peewee and the chickadee seem to be very versatile. So a couple things happen with these patches, right? When we cut them, when we fragment them, when we build a highway, when we build a housing development, we isolate those patches, so it's harder for animals and plants to get across to the next one. And we also might be changing the shape of it, so the core versus the edge ratio can shift. And the more isolation, think about the genetic implications to that, right? Imagine you had two oceanic islands really far away, and then some close. The ones that are closer, there's going to be more gene flow. The ones farther away, it would be harder, and you probably only get species that can fly strong. They're strong flyers or can swim well. And so any way that we can connect those patches, if they get isolated, would be great. And that's what we talked about with hedgerows, the stone walls with the vegetation that grows alongside of it. Again, they're linear, so they're going to have more edge, less core, but there's still resources they provide and a little bit of cover as the animals are moving across that spot to get from one patch to another. So it's not ideal, right? There's some pros to it because gene flow happens, but there's also some negatives that we talk about. One of those negatives was nest parasitism, higher rates of nest parasitism when you've got a smaller patch, which again would reduce your core and increase your edge. And the big overlying theory, just like when we talked about communities, it was disturbance theory, right? Medium, moderate, intermediate disturbance theory. That was the really important one for communities. For landscape, it's island biogeography theory. And these researchers really just wanted to say, hey, can we kind of have this hard and fast rule? Can we estimate what our equilibrium number of species on a patch or an island would be, just if we knew what the area of that island was? Do we assume the larger the area, the more species? Can we make those big, broad assumptions? And so MacArthur and Wilson came up with this idea. At first, it was about, they did their experiments. These were like based on real life experiments. So if any of you guys have been down to like the Florida Keys or the Caribbean islands where there are mangroves and you've gone kind of kayaking around there, imagine a bunch of like an archipelago of mangrove islands off of the coast, right? Some are going to be smaller and clustered close to the mainland, and then others will be larger, maybe they're farther apart, but they're a bunch of islands of different areas. They have bugs all over them. And so these guys, it's crazy. This is something that never would fly these days. But these two researchers went out there and they put tarps over them all. So they got a sense of the area, they maybe mapped the size of that island, then they tarped them, and then they pumped this nasty insecticide and basically fumigated all those islands, and then removed it and observed the response of recolonization. So the immigration colonization concept that we cover in this topic. And so they watched over time the communities rebound. And what they found is time and time again, the number of species that came back was about the same, but the type of species differed. So it didn't really matter, and likely it was, a function of how far away it was from the mainland. Probably the ones that colonized farther out were species that were stronger swimmers or flyers, right? But time and time again, it plateaued at that same equilibrium number of species, maybe because of the size of that island and the isolation effect. So again, that's where that information came from. I wanted to just throw that out there for you in case you've ever kayaked out there and seen them. Pretty cool to see. So, there's turnover all the time in these communities. And we know that, things move back and forth across the landscape. So there's immigration, and extinction happens all the time. We've probably heard of, again, local extinction. We don't have mountain lions in our area anymore, but we used to. So mountain lions are not gone completely. The species is not gone. We see them populations in very across the country, just not in our area. So that's what we would consider to be an extirpated, so locally extinct population. okay? It's found elsewhere across the country, so it's not extinct, but our local population is extinct. So we call that term is extirpated. So they were really, interested in seeing how these islands wink in and out, what the equilibrium number of species is. And so they basically said, all right, what if we looked at extinction rates and what if we looked at immigration rates? And we used those two rates to estimate, where the two lines meet, and that would be our equilibrium number of species. So the idea is, at first, it's an empty vacant island, so everybody can come, right? Anybody who can get there can come. So you'll have high rates of immigration early on when we've got kind of a vacant island or an island that just formed, a patch that just formed. And when we think about extinctions should be really low at first because there's not a lot of people or organisms on there, but over time they lose resources as more and more kind of join in, right, and enter that system. So we would expect extinction rates to increase over time. And where those two lines meet will give us our equilibrium estimate number of species that can be supported on that island or that habitat patch. We can change the slope of those. And imagine changing just one slope and what that would do to that meeting point. It'll shift it either right or left on that x-axis, right? And so if we could somehow reduce our extinction rates or increase our immigration rates, then we would benefit and have more species that could be on that island, okay? And so we have to think about, well, what would allow us to reduce that extinction rate? How could we lessen or slacken that slope right there, that rate? Well, we could make it a bigger island, okay? So, over here, if we change the size of that island, not that this really ever happens, like in an ocean, but maybe on land, maybe your island is agriculture, right? And you would then just cut down forest to make that agricultural patch bigger. And this was good for meadow mice. I'm just making this stuff up. And if that was the case, we could slacken the slope of extinction and it would shift our species, our equilibrium species, to the right. Okay, that's what we would do to reduce our extinction rates or, lessen the extinction rates, is we would have a bigger island. Okay, and then in the opposite, well, of course, there's examples of this. If we want to, increase species, and we're thinking of isolation of islands, how far away they are from the mainland, if we could ramp up immigration or colonization, that's going to shift the slope up, and it would shift our species to the right as well. And we do that by making it a closer island. Okay, so scooting it over. Somehow connecting it so it becomes one larger patch that's closer to that mainland source would be a good way to do that. So some of this stuff is very theoretical. You may not imagine that this could really happen in real life, but if you were planning to manage a certain species or conserve a certain species, you can imagine if you want more of them, then you want a bigger size patch and you want it closer to where more of them are, right? Those are the big take-home messages. So very important. If I were to ask you guys, what are the two rates that are important in island biogeography theory? I want you to say extinction and colonization or immigration, right? And when I ask you, well, what factors can shift the rates, influence those rates, that's when I want you to say, well, for extinction, it's the size of the island, the area. And for immigration or colonization, it's that isolation or distance to the mainland, okay? So keep those straight in your head. That's a very important concept. And again, across species, across taxa, we see it with butterflies, we see it with wetland species across, animals and plants and mammals. The bigger your area, the more species that you can accommodate. The more isolated your island, the fewer, mammals that you'll have. And we saw that again with our lizards in those Caribbean islands. And this was just a recent article, sort of more recent article, talking about how area does sort of, the size of your island does play into extinctions, really across the board, across taxa. That's the big take-home message from this visual on this slide. Okay, connectivity, as we mentioned, really important. If these patches are isolated, we got to connect them somehow, whether it's a hedgerow or not. That's going to increase gene flow, less inbreeding, less genetic drift. But the bad stuff might outweigh, it really is very context dependent, it might outweigh the good stuff. So more disease, more harsh abiotics, fire, those nest predators, mesopredators that love these linear features. And so, you know, we talk about this idea that we have a really fragmented habitat. Like think about, you know, if you looked at aerial photos, for those of you that are in GIS or remote sensing, if you looked at aerial photos from say 1920 of this area versus now, or, you know, any urban system, you would totally see how it's become much more of a patchwork. Our patches are smaller and more isolated, and that has implications for the plants and animals that are living there. And so when you think about designing a way to protect these organisms, if their numbers are low, if their populations are declining, we really can't say, oh, you need to have a 5 million square mile, you know, patch. And that way we can bring polar bears back down into, you know, the lower parts of Canada, maybe into the Adirondacks. I mean, no way, Jose, right? What we have to be is realistic, and we have to say, well, this is what we've got. We've got a patchwork, a mosaic. We will probably be able to manage the population and they'll do okay and won't go locally extinct if we can connect those patches, okay? So building overpasses or underpasses or culverts, fencing them along a river so that they don't get, you know, out of the way, and know that they can connect by following that river corridor. And if we at least allow connection across these patches, we can consider all 27 patches or however many patches there are as a meta population, okay? As long as gene flow happens, it's okay that some of those patches will go extinct, others will do well. And back and forth, they'll move across. The ones that are doing well will have more births, and they would be called source populations. The ones that aren't doing as well, maybe that's because they just were a source, and now they're kind of regenerating. And so we've seen emigration out of that patch that once was good, and now it's recovering, and we would consider it to be less quality. Maybe less births would happen on that patch now than before. That would be a sink population, a sink patch. So when I say meta populations, I want you to think source and sink. In a source, you tend to have, you know, better scenario, more births happen than deaths, higher quality habitat, maybe it's a little bigger. Sinks would be lesser quality, maybe more deaths than births, but it also is an opportunity that you could bounce there briefly while your other spot is recovering. And so there's going to be sort of this movement, and again, very important that these patches are connected, okay? So keep these things in mind. Meta populations are facilitated by gene flow and immigration. This is very much what is going on with these owls out west, right? So the owls really need like long swaths of old growth forest, but that is in like direct, you know, they're fighting with the timber companies, right? They want that old growth timber. And so if lumber companies win out, we end up with these patches of different sizes, very much isolated, and these owls that are in decline. And so how can we help them out? Can we connect their patches somehow? And you can see that this could be a metapopulation scenario with these owls. We also have to be aware of the competitors that are moving in from the east, and that would be our barred owls. They're a bit more aggressive, and so we may have to do some barred owl relocations in these areas too to help them out. It may not be just good enough that we connect these patches or have, you know, little stepping stone islands of older growth to get to those bigger source patches, like the one that's got 190 and 266. Those would be sources, whereas some of the lower ones would maybe be sinks. Okay, so time and time again, the patches that tend to be occupied are the ones that are less isolated. Okay, so the less isolated you are, the more occupied, and the bigger the patch area, the more occupied. Those below this line are the ones that are unoccupied. They are farther away and they have smaller areas. And that's true for shrews, So our smallest of mammals, right, these guys have that high metabolism, but it's also true for our invertebrates. So a lot of our butterflies, including the Karner blue, which is down in the Albany pine bush, behave this way as well. So I did mention these assumptions to the models. I'm not going to dwell too much on that. Getting back to where we were at the very end here, think about this in terms of if you were a landscape planner and you're trying to plan for a new housing complex, and you've got conservation area around you. It would probably be good for you to plan a development that doesn't have 15 different driveways, really long driveways, right? There's a lot of edge and it would fragment those patches. So oftentimes, you know, if you think about these island biogeography concepts in another discipline, they can become really helpful in serving a couple of different, you know, purposes. And that would be, you know, you've got a nice housing complex that has good flow, but you also have protected some of those areas that were very kind of sensitive. So, you know, definitely important for you to map out first where big patches are, patches that you can imagine would be highly prized for the animals. So in Boston, so I live outside of Boston where I grew up. If you've ever been to Fenway Park, a little bit south of Fenway Park is the medical district, a couple blocks. And if you were to look in an aerial image, along the stream that runs through there is this, they call it an emerald necklace of parks. And Olmsted, the famous park designer, created these. And so he connected these patches and then linked up some of the cemeteries that have a lot of trees. I mean, it's really beautiful. Knowing that these would be like reserves in this urban setting. Hold on, I'm gonna shut the door, the kids left it open. So find those areas, target them for conservation, don't build on them, and make sure they're connected as best that they can so that they can serve as corridors. Anytime you've got the natural habitat, obviously don't, you know, rip it up. Do some plantings of native vegetation. And have some habitat heterogeneity. We've seen time and time again that that is actually really good to gain the most diversity in a system. And so here's an example, as I mentioned earlier, with like housing developments. So, you know, this is in North Carolina, and this stuff around here is the habitat, and this is your potential area that you want to put your development. So how do we set it up? Let's have one long linear road here, and then let's build a cul-de-sac with, you know, driveways that are super tiny that are really just around that. And you can kind of preserve something in the middle here. And maximize the amount of green space retained. But it's, again, that patch dynamic connection. Think about shapes of patches, size of them, and reducing edge. Again, a circle is the best shape for you to have to reduce the amount of edge versus core, right? And that's really important to consider. So with landscape ecology, it's an applied science. It uses technology. It uses these theories, and it helps link communities to what we have done to the landscape. It's jumping us up to ecosystems shortly. We are going to measure lots of stuff: edge, contagion, the arrangement of those patches, how big our area is of the patch, and isolation. We're going to think about connecting them with these hedgerows and how it can be good, but it can be bad, and you have to kind of weigh that out species by species. Very important, again, to commit to memory the island biogeography theory, thinking about the two rates that we use to get that equilibrium estimate of species, and then how we shift the slope of those two rates with either adjusting the area or the isolation of those patches. And think about metapopulations. It's a concept. It's a concept that we have to deal with now. As long as we can connect our patches with gene flow, then we have a metapopulation, not necessarily a whole-on old-fashioned population, but we have little populations, subpopulations, and as long as they're connected, we can consider it a population, because genes are flowing across these smaller patches, okay? And that term source and sink is in there. And then how we can use this for planning with housing and stuff like that, okay? Any questions on landscape? All right, sounds like we're good. Now that we know we've got these landscapes, now let's think about how energy moves across that landscape, okay? Energy in the form of our plants, in the form of our animals, and they all have a given lifespan. When they die, they decompose wherever they might be and return those nutrients back to the soil, okay? So we're gonna be thinking about nutrient flow across our biomes. So in the end of this lecture, I'm gonna give you an example of nutrients that are maybe limiting. You know, if we were thinking about photosynthesis in a marine system, so the ocean versus a freshwater system versus, you know, on land, is there a nutrient that is such that if we don't have enough of it, everything shuts down and productivity is really low? It varies actually in those different biomes, terrestrial versus marine versus freshwater, okay? So what I want us to begin with is this visual of productivity coming from our satellites. And so you can, you know, have satellites that are set up to look at chlorophyll levels, and you can see where it's brighter would be a patch that maybe is, you know, is eutrophied, versus where it's cooler would be maybe an area where you don't have a lot of nutrients available to you. And so in the middle of the ocean, we call those dead zones. There's not a lot of productivity. But imagine, and I sent you guys an email right before I got on here, but imagine if you could do some kind of crazy engineering and dump a whole bunch of whatever nutrient is limiting in the ocean, in those dead zones, and imagine the algae blooms, right? And these little algae machines, green algae, blue-green algae, cyanobacteria, just suck that carbon in, carbon dioxide coming in, right? And sequester it. And think about what that would do to the carbon exchange that we have, right? Not to say this is awesome. There's definitely, you know, research being done that shows, I don't know, there's a lot of like bad things that happen in terms of like a worse gas being emitted, like a nitrous oxide being emitted, that is a worse greenhouse gas than carbon dioxide. So, but just knowing that there are researchers that are out there trying to finagle ways to maybe assist in enhancing production in areas where it's lower is pretty awesome. And using technology like remote sensing so we can see it across the globe, not just at that fine scale, is really important. Now remember, way back when, if you were trying to estimate productivity, you know, you're probably doing it with a plankton net behind a boat, and you're looking at it, you know, in certain small areas in your lake. Maybe you're doing it on a farm field, and so you'll burn, you know, some of the crop residues and get a sense of, you know, what the energy was in that burn, and have a sense of productivity. But using this technology is pretty awesome because you can see it across scales, right? A broad scale. So when we cover ecosystems, we're going to define it. And basically what an ecosystem is your community, the plants, the animals, and how abiotic factors play into that. So soil, nutrients, moisture, temperature, sunlight, all of that stuff, very important to evaluate how that energy flows across the systems. And so, you know, we're going to think about how that productivity, how much plant will grow, can vary according to what biome you're in. And those biomes that we're going to look at are the marine system, the saltwater oceans, our freshwater systems, lakes, ponds, and then the terrestrial systems just broadly. And so we'll think about the things that, you know, alter net primary production in those systems, and then think about how that energy is flowing through that pyramid of energy that we've got with our producers, herbivores, and our predators up at the top. So here we go with our ecosystem, right? We've got our community here, our biotic community, and we're going to then bring in that abiotic component. How are those nutrients flowing through that system, through that community? So we'll talk about, you know, fluxes among the pools, the fluxes of nutrients among the pools, one pool being the primary producers down at the bottom. Remember when we talked bottom up, top down. And then our secondary production would be the herbivores, and then our tertiary would be higher on up, right? And again, scale, level of detail, all that stuff comes in. But as that energy moves up the system, that's what gives us the shape, right? That's why we have a lot at the bottom, highest amounts, more individuals, and greater energy. And then some of that dies and doesn't transfer to the herbivores, right? Because some of it has nasty chemicals in it, right? Allelic chemicals that they don't want to digest. So the plants that they don't eat, those herbivores, return to the decomposer, pool, right? But the ones that they do eat, become more babies of herbivores. And then if there's enough of them, then the predators have enough food and we'll have a nice predator part of the pyramid. Okay, so we mentioned there's a level to how high our pyramid of energy gets, and it usually caps out around 4 to 6, right? So again, primary production is the autotrophs, the plants, the cyanobacteria, the things that are making their own sugars. Secondary production, these are heterotrophs, consumers, decomposers, multiple trophic levels. I mentioned them as herbivores, but it can be anything that's eating those autotrophs. And we're going to be seeing how that energy, from the form of plants moving into herbivores, moving into predators, flows across the systems. And we'll do that by looking at biomass. Very easy, right? Imagine you took the standing crop weight, you harvested your plants, you got a weight, you did it at 2 different time periods, you subtracted them. We're going to look at how, photosynthesis and respiration, where they kind of meet, and what that means for net primary production. And so the term gross versus net, think about your take-home pay versus what happens after taxes, right? So when we are looking at the amount of energy or carbon in a system per unit time, we're looking at photosynthetic rate. When we look at net primary production, we're subtracting off that respiration, okay? But the gross would be, we're including, and then we're subtracting off respiration for the net. And so, again, looking at biomass at time 2 minus biomass at time 1, that would give us a sense of the energy in that crop system, right? Just a quick and easy weight. And you can burn that material and get an energy reading. You guys might have done that in like a chemistry class. Now, Mary Aldred does a lot of research that has to do with energy uptake, nutrient flux in wetland type systems. So she does some really cool things. She has machines that will allow you to evaluate the amount of carbon uptake during certain scenarios, whether there's oxygen in the sediment where this is occurring or not, whether it's night versus day. So let's just take a look here in the dark. Okay, so if this is nighttime, or you have a bottle that's covered in like brown or something dark, so no light is coming in. If you're measuring carbon dioxide when it's dark out, or when your container that you're doing this in the lab is covered in black, then the slope that we see here is respiration rate. If you have it open to the light, if you're doing it during daytime, or again, you have a clear container with your plant inside and you're looking at carbon dioxide flux, here you're going to be looking at photosynthetic rate. So that one that's in the light would give us our net primary production. And here we can evaluate gross by subtracting off our respiration. So again, just kind of talking a little bit about what Mary does. Wanted you guys to see it, but she's got some cool visuals here. You can have a leaf in a chamber, and the chamber can be dark, chamber can be light. You can do it in an aquatic system, so you can have water kind of penetrating through in these aquatic systems. And anywhere where we have light that's kind of getting in there, we're getting a sense of photosynthetic rate, and anywhere where it's dark, we're getting a sense of respiration rate. And we can, again, as I mentioned, measure energy flux, carbon uptake, photosynthetic rate, all that kind of stuff using remote sensing. So again, that technology, we're using it to our advantage, and it's letting us look across scales, across the globe. And really, we're just tapping into that satellite being able to pick up the chlorophyll A spectrum. It's getting the reflectance there. And like I said, it can save us time, like dragging a plankton net on a boat and then bringing the sample back and putting it in a spectrophotometer, which many of you guys might have done in a chemistry class. But again, it's going to be small scale. It's not going to be scaled up like this can be. And so again, remote sensing, satellite use is going to allow us to look at a very broad scale the amount of energy that we would see in a system. We'll be able to pick up that there's those dead zones in the ocean, and along the coast is more productivity. We'll be able to see there's way more productivity in the tropical rainforest biome or the temperate rainforest out in, say, Oregon, compared to maybe the desert biome. So there's a way for us to kind of evaluate that. And so we can look at, again, net primary productivity. If you were doing GIS, you might have seen there's a layer available to you. It's called the Normalized Vegetation Index. And really, it's just you know, a calculation that takes your near infrared value and subtracts off your visible light value for that cell. And then the denominator is just adding the two. And that value gives us the level of vegetation that you have in that pixel in your GIS map. So an NDVI of 0 would be a barren spot, and one that is 1 would be that there's a lot of productivity in that particular system. So I just, again, wanted you to see how they arrive at that value. So remote sensing, super, super important. Again, when we have high rain events, when we have a lot of runoff, when we have eutrophication occurring, we can pick this stuff up in the satellites, and that can be helpful, especially, you know, when we're talking about saxitoxins. If you guys have heard of, you know, those red tide blooms that can be very harmful to our dogs as well as to us if we ate the shellfish. We can maybe track when that would occur. We can block, you know, access to the beaches and suggest people really take it easy and don't go in the water, don't go fishing, don't eat the stuff during that period of time. And as I mentioned, primary production, the amount of energy entering the system, you know, via plants, can vary across our biomes. So here we have terrestrial biomes, here we have our aquatic biome type situation, and things that can alter, just like we talked about things important for biomes, temperature, precipitation, and soil, right? So temperature, moisture, precipitation, temperature and light, and then nutrient availability, right? These are the combos that are required for us to think about net primary production, or how energy can flux across these biomes. You think about the nutrients, macro and micro, that are limited in these soils, and then the water, and then, you know, sunlight potential, and temperature, which if it gets too hot, you're not going to have, you know, much of that happening, because you get that photo inhibition point. But just take a quick look up here in the terrestrial system. We've got our tropical rainforest, tropical seasonal forest, temperate rainforest, boreal forest. So most of the forests have high net primary production. Extreme desert, tundra, those extremes that we talked about where stress-loving plants would be, have much lower NPP, not a surprise. Swamps and marshes, estuaries are important, coral reefs, salt marshes, high production. Less production, those dead zones in the open ocean, okay? So if we could somehow relieve that nutrient stress, what nutrient is limited to them, then we can ramp up the amount of production in those systems, which can be really important. Okay, so again, what happens in terms of primary production? We're looking at NPP on our y-axis and precipitation. It does plateau. You can have too much rain. If you get too much rain, you tend to get rot, right? Fungal growth, bacterial growth. And you can have too high a temperature. Photo inhibition can occur. But for the most part, moderate levels will maximize your NPP. Now think of an ocean system, right? Or any kind of a freshwater system, right? Your organisms that photosynthesize are up in that photic zone. So you have a limitation for net primary production that goes as far as that light will penetrate into that water, right? Right at the top, you might have a situation where the UV light is too harsh and you get photoinhibition, but just down a smidge is the happy zone for these organisms to be able to photosynthesize, right? Anything deeper, you're going to run into problems with not having enough light, the conditions change. But again, there's that little window there where there might be a little photoinhibition. So secondary production, again, how is that energy moving up the food web? We know that there are way more individuals in our bottom, primary producer level, bottom up. and then there should be fewer individuals. That's why the slope tapers on the triangle, right? Fewer herbivores, and then there'll be fewer, even fewer at the top of that pyramid. So in terms of n number of individuals, size-wise, plants are way smaller individuals, right, than herbivores. And herbivores may or may not be smaller than, say, a top predator. But in terms of energy flow, there's less energy at the top than there is at the bottom. Okay, so if we want to kind of see how this energy is flowing from plants on up to herbivores on up to predators, we can look at trophic efficiency. Okay, it's basically the net primary production of one trophic level divided by the net primary production of the trophic level right before it. Okay, so secondary production, again, once the plants have been captured and moved up the food chain, we're talking about how it's captured into herbivores or how it's captured into predators. That's secondary production, whereas primary production is the energy captured into the plants, right? So we can set up a trophic efficiency that would be the NPP of, say, the herbivores divided by the NPP of those primary producers. And that would give us a sense of the energy in that system. As I mentioned, energy transfer gets reduced on up that pyramid, right? And so 2 to 24%, with an average of about 10% of the energy available in the level below, moves up. Okay, so you've got a lot of waste. A lot of it enters the detrital pools, right? And we'll talk about that shortly. So you can see between 2 and 24 instances where we would see it moving up the food chain. And on average, the transfer efficiency is about 10%. So at each level, depending on how high your pyramid is, you should have 10% less energy in the pyramid level above it than you did in the one right below it. Okay, and that's what gives it that pyramid shape. It's not super efficient, but certain systems are more efficient than others. We'll learn that aquatic systems, more gets transferred than the ones below it. Okay, and so here we've got production, the producers, right? And you can see that they respire, and some of the producers, the plants, will move up into the herbivores and they respire, and some of those herbivores will move up into the carnivores, right, those predators, and they will respire. But some of the producers, a lot of it actually, don't get eaten by those herbivores, right? They just can't process it. So it enters this dead organic matter or detrital pool. Okay, every single one of these guys dies and decomposes and adds to that detrital pool. Okay, and the microbes will add to that detrital pool, right, as they start breaking stuff down. So again, think of how energy moves through the system, through the pyramid, then everybody also is dying and adding to that pool at the bottom of the detritus. Okay, so that's important for you to consider. And here's just a visual of that production kind of moving forward, right? Net primary production eaten by those herbivores and predators. They take on that energy and then they make babies, and they will ultimately die and return that energy into the system, okay? I'm just gonna finish off with this last take-home message here. I mentioned not everything transfers across those biomes efficiently. And we mentioned, I think we've seen this slide before, but in aquatic systems, there is a lot more transfer of energy up into the herbivores. More of it gets eaten because the water helps support the structure of those plants. And so they don't have to have as much of those tough lignin and structural components in them. So more of it gets eaten. Whereas on land, a lot of that primary production does not move up to the herbivore pool because it's distasteful, because it has those harsh kind of chemicals and structural components that are required to deal with the pressure of gravity. And so a lot more on land material, like our plants that are on land, enters the detrital pool instead of moving up to the herbivore pool. In a marine system, in a freshwater system, way more goes into the herbivore pool than goes immediately into that detrital pool. So it's important for you to kind of consider that. So in aquatic systems, we have way more grazers and herbivores than we do in our terrestrial system. In our terrestrial system, we have way more decomposers right off the bat, because not a lot of those plants get consumed, okay? The next time that we meet, I'll talk about nutrient limitation across the marine, freshwater, and terrestrial systems. We'll finish off with that, and we'll do a little bit of review, okay? Sound good?
Sounds good.
All right, we'll see you guys on Wednesday. Have a good one.
See ya.
Bye.